Population improvement methods, and we really have only two left to cover, which you're probably thinking with a sigh of relaxation that, man, finally we're getting to the end of that, because so far I think we've covered 8 of them. In fact, we covered a lot of them the last time, so I acknowledge that you might have been confused by some of those, but we will take time to review them in a little ways through the period today. But what I'd like to do is to cover the S2 intra-population improvement method, and then to transition into inter-population improvement and to talk about the half-SIB reciprocal recurrence selection. So we're going to cover those 2 methods, and then we'll follow it up with some review. I've got some concept maps that will help us to kind of drop back and look at the big picture once again from a bird's eye view, and to use that as a time to review. So that's kind of where I'm heading, and we'll see how far we get with that. But I do know that we'll get through all of population improvement and be able to have a little bit of discussion at the end in terms of things to think about. Now that you've had 10 population improvement methods, how do you decide which one to use in the particular circumstance that you're in? So I'll give you 4 criteria to think about. There's never a hard, fast answer. 2 breeders might come up with 2 totally different methods that they want to try to use. But at least these are criteria for you to think about. And none of those 4 criteria will be surprises by the time we get there, because they're really the same criteria that we've been talking about all along. So as we go through the class, again, as you have questions or need clarification on any of these methods, especially as we go through the review, please slow me down, and I'll try not to go at quite as fast a pace as I did the last time, but let's try to take some time to really understand these. And although you don't have an exam on these, again, I want to make sure that you have sufficient enough to review so that you understand all of them. Now, the last time we ended up talking about the S1 progeny test, and just to review that very quickly, because I want to relate that to the S2 progeny test in just a moment. But again, you don't need to make any, allow open pollination to occur, because you're not evaluating half-SIB progenies with this particular test. But you do need to go in by hand to make S1 progenies, do the selfing. In the second season, you're growing out replicated self progenies, S1 progenies, and basing your evaluation, if it's yield, you're taking the yield on those progeny rows and basing the evaluation on the S1s. Then you take remnant seed. Now, does everybody by now understand the concept of keeping some remnant seed? Okay, I want to be sure you understand that, because I know in some previous years people have been confused with what I'm talking about with remnant seed. But take some of that remnant seed out of seed storage of those same S1s that were the superior ones that you've identified in season 2, then grow those out in season 3 and allow those to open pollinate so that you can get your genetic recombination. And we said that one major advantage, a few major advantages in fact, of this method is that, the principal one is that there's more additive genetic variation among S1 progenies than there is among either full SIB or half SIB progenies. So that becomes an advantage, because that impacts the A value in the numerator of your response from selection equation there. In other words, that A value becomes 1, which we haven't seen since we talked about phenotypic selection a few periods ago. So that's the major advantage. Plus, by selfing, you might be able to uncover some deleterious recessive alleles there in the population faster than you might uncover those by just random pollination. So those are 2 major advantages. Does anybody have questions on this method before I extend that to talk about S2s then? Everybody understands that pretty thoroughly. Okay, unfortunately, I've never made up a schematic for the S2 progeny methods. In fact, you may or may not have noticed, but if you look up here in the right-hand corner, you could probably tell that these schematics are pretty old now. They were made probably close to 25 years ago, when a few years after Ronnie Kaufman actually came to the department. He was actually teaching this course, or the predecessor of it, and he was the first one that asked me to teach the section on population improvement. And those were back in the years when not everybody had desktop computers. In fact, he had the first one in the department, which was a predecessor of the Apple Macintosh. It was called, I think it was called Elisa. So that was the first one that was in the department, and he told me that if I could write down on a piece of paper what this schematics look like, then he would get on his computer and make these up. So that's why you see both mine and his initials up here, and even the former number of the course. So these do go way back, but they're still very valid schematics to use. But unfortunately, I've never made one up for the S2 progeny method. But it's pretty easy to extend it, because essentially you do the same thing. You start out by making S1s, but instead of in season 2 growing out replicated S1 rows, you only need to grow out really one S1 plant per parent plant that produced those S1s. Now I would plant out more than that, really, maybe 3, 4, or 5, just to make sure you get S2 seed, sufficient F2 seed, okay? But it's really like just like you've had single seed descent in this class already, right? It's really identical to single seed descent, but you're only carrying it down 2 generations instead of maybe 4, 5, or 6 generations. So you really come down to season 2, and instead of growing out a row, again, you grow out just a plant that represents each one of these parent plants, and then self that to produce an S2. And then in the next season, in season 3, you're growing out replicated S2 progeny rows. Evaluate those. Keep some remnant seed of the S2s, just like you did the S1s in the previous method. And then you take the remnant seed of the superior S2s and grow those out in season 4 and allow those to interpollinate to get your genetic recombination. Does everybody see that then? We're just extending it down one more generation there. But you have to do it like single seed descent. I had this question in my mind a few years ago, soon after I started teaching this. And I've never seen it described in a textbook anywhere where they said how you do this, carry it down to the S2 generation. But you do not grow out an S1 row and self a whole bunch of S1s and then balk those that represent an S1 progeny. You do it just like single seed descent, or this method doesn't work very well. Any questions then? Everybody understands how to do this? Okay, now the major advantage, now let me flip to where my slides are here to catch up. The major advantage is that in contrast to the S1 method where the A value was 1 up here in the numerator of the response from selection equation, now it's 1.5. In essence, at least theoretically, you've created additive genetic variation among the S2 progenies. You have more than you had even compared to the original parent plants that you started with in the plant population. And that can be a real advantage, especially if you have a very low heritability for a trait, is to actually create more variability among the units that you're evaluating. And so if the more distinctiveness, genetic distinctiveness or variability that you have among the units that you're basing your selection on, the more chances that you'll be successful as a plant breeder of identifying the superior genotypes. Now notice that we also have this beta in here, but now it's beta 2. It's just like the S1 progeny method, but beta is a little bit different equation, but it's still there only if there's some dominance for the trait. But again, like I said the last time, it is not the dominant genetic variance. But if there is no dominant genetic variation, then beta 2 will be 0. And again, this is divided by the standard deviation among the S2 progenies for the trait that you're evaluating. Okay, so that's the major advantage, is that this becomes 1.5 up here for that A value. Another advantage, I don't have it written here, but just like the S1s, again, you can uncover some deleterious recessive alleles and discard them from the population. Even easier among S2s than you did the S1s. But a third advantage is this one, and that is that if you're working with a crop like corn or any crop where you eventually will be producing inbred lines and crossing those to produce a hybrid variety, if you happen to be looking at S2 progenies and you see one that looks really, really good, I mean just outstanding, you're already part way to developing an inbred, and you can just carry that line on down a few more generations to produce a superior inbred line. So it does give you that opportunity because you're already part way there. Okay, and again, like I've said, you never add an advantage or make a modification to add an advantage without some disadvantages. And again, like some other genotypic methods, it is a lot of work to do this. in that now you have 4 seasons of work to do. You have to make S1 progenies and then S2 progenies, both of those by hand, and then evaluate the S2s, and then season 4 you're interpollinating remnant S2s there, from remnant seed of the superior ones. So there's 4 seasons of work to accomplish, and there's no way that you can get 4 seasons into 1 year, so this is actually going to take 2 years to accomplish it. So then you have to weigh out, well, what's the progress from selection per year, per unit of time there. Yes, Andres?
So in the first season, the parents open pollinate?
Nope, you don't need to open, in the first season there, you said?
Or
okay.
The parent plants, the original?
Yep. You don't need to open pollinate those at all. So if you're working with corn, you don't need to be concerned about prolificacy because you only need 1 ear and you're going to self-pollinate that ear. So no open pollination, you just self-pollinate it. The only time there's any open pollination is in season 4 where you're growing out plants from remnant seed of the superior S2 so that you can get genetic recombination. Good question, good question. Any other questions or comments that you have? Okay. So that's really the last of the intra-population improvement methods, where you're trying to improve a single population at a time, although you might have a whole bunch of them in your program at any one time that you're trying to improve for different reasons. But now I'd like to morph into talking about inter-population improvement. Inter meaning between populations. On this one, you're going to be trying to improve 2 populations simultaneously. They're in parallel programs. You're working with them at the same time, same traits, and you're trying to improve them in such a way that once you cross material from the 2 populations, that they will nick real well to give you hybrid vigor. So the objective, the breeding objective of interpopulation improvement is to result in a hybrid variety. If you're not producing a hybrid variety, you really wouldn't use this method. Okay? So again, you've got that objective, again, to improve 2 populations simultaneously so that the gene actions are complementary. Again, your objective ultimately is after you go through a few cycles of interpopulation improvement to extract material out of it, whether they're ultimately inbred lines or non-inbreds, but whatever it is, you're crossing plants from the 2 populations to produce some sort of a hybrid variety. Okay, let's see how this works. Now, we're only going to cover half-Sib reciprocal recurrence selection. There is such a thing as full-Sib reciprocal recurrence selection, but we're not going to cover that. But you understand what full Sibs are, and I bet you can figure out how to do it yourself after you go through this particular method. Now, this looks kind of complex as it is up here on the screen because there's just a lot of stuff there. But what you need to do is to first of all look at one side before the other, because the right side is really a mirror image of the left side. Okay? And if you were to look at one side at a time, you might notice that it's exactly identical to a method that we've already covered. And that is the half-Sib progeny test using a tester. Except when we did that, when we talked about that during the last period, our objective was to determine which parent plants were homozygous dominant and which were heterozygotes by crossing it to a recessive tester, a poor performing tester. Our objective now is not to do that. I mean, it's exactly the same method, but we're going to use a different kind of a tester because now our tester is actually going to be the population. For instance, on the left side, we're trying to improve population A. On the right side, we're trying to improve population B. The way that we're going to improve population A, ultimately what our objective is to take plant material out of A and cross it on to B to produce a hybrid variety. So we're going to try to improve both population A and population B so that they nick well together. And the way we're going to do that is we're going to improve A by crossing it on to B and identifying the parent plants that produce the superior progenies when crossed onto B. On the right side, we're going to improve B in such a way that it nicks well with A, so we're going to use A as the tester population. So in this case, the tester populations are going to be good performing populations, ones that we're actually going to use to develop a hybrid variety. Now let me hesitate there and ask if you have any questions so far before we dig into the details. Everybody see the big picture of what we're doing with it? Okay. So let's look at the left side first. With the left side, again, we're improving population A. That's our target population. And we're going to use B as the tester. So what we're going to do, in contrast to what this says, which I wish I had the original files where I could go back and change it, you could do it as it says here, where you're using A as a male and B as a female. Really the best way to do it, the easiest way, is to go into population B. You know, you have to be growing these at the same time in fields that are pretty close to each other. And go in and collect pollen from a number of plants of B, just a collection of pollen, and block that together. And then go over to population A next door to it and pollinate individual plants in A there. So you're producing a half-sib progeny. If you harvest a seed on each A plant, it's a half-sib progeny of a bunch of pollen from B onto each plant of A. In season 2, then you would need to plan out your replicated half-sib progeny test, identify the superior half-sibs. Then let me ask you a question. Why would you not want to take remnant seed of the superior half-sibs and use that as your cycle 1 seed? And it's not the same reason for not doing it when we talked about half-sib progeny test using a tester when we're using a poor-performing tester. Now it's a different reason. Yes, Nick?
You don't want B genetics, population B genetics and population A. You want to just retain population A genetics, genes that are like good to combine with population B as like separate.
Yep, that's exactly it. You do not yet want to mix up A and B yet. When you get down here to cycle 1 seed, you do not want it to be A and B, because if you go through 3 or 4 cycles of selection, before too long A and B will be the same thing because you're mixing both of them up together. It'll be the same population. And yes, you could do that. As a breeder, you have every right to do that if you want. And it's not like you can't take that overall big population that is now a mixture of A and B genes and find 2 plants that will nick well together with each other to produce a hybrid. But your chances of finding 2 that will nick together are going to be much, much harder to identify if you've mixed up all of those genes together than if you kept those 2 populations separate. We look at that mathematically in the quantitative genetics course in about the 2nd week of class there to help you understand why that happens. But you need to keep the 2 populations separate until the very end, until you're ready to extract material out to produce that final cross to be the hybrid variety. Otherwise, it's hard to find plants that will nick well together to do that. So you're just using B as a test population with A to identify the superior A plants that will cross well with B. But then, to keep the population separate, you'll take you have to, back here in the season 1, actually produce S1 plants, S1 progenies as well, so that you can take the S1s of the plants that produce the superior half-sib progenies, grow those out in season 3, and allow those to open pollinate to get your genetic recombination. So your cycle 1 seed down here is just going to be genes from the A population. Will not have any B in it. Everybody follow me on that? Yes, Andres?
Are these 2 selections done simultaneously?
Yep. So at the same time you're doing this over here on the right. You're doing that simultaneously. So let's look at the B population and improve that. We're going to do exactly the same thing, but now we're taking a collection of pollen from a number of A plants. So you have to have A and B populations growing out at the same time. You know, you don't want A here in Ithaca and B up there in the North Country. You want them both right there in Ithaca, probably right there in the same field. You know, not real close together, but close enough so that you can walk back and forth there between the populations. So a collection of A pollen, cross it onto each one of these B plants, harvest the B plants individually so that they half-sib progenies. Season 2, you're growing out the half-sib progenies. You know, again, it's B times the test population, which is A now. Identify the superior ones. You also have to have S1s that you're producing up here so that you grow out the S1s that came from the parent plants that produced the superior half-sib progenies. Again, your cycle 1 seed is going to be only B genes in there, no A genes. But hopefully B plants, B genes that will nick well with these that over here are identified as superior genes in the A population to give you a better chance of finding a superior hybrid variety in the very end. So you'll go through this process, maybe a few cycles of selection, 2, 3, 4 cycles of selection. Again, A and B being separate populations and using each other as test crosses, tester populations, so that when you get to the final cycle of selection where you feel satisfied that you've improved the populations, then extract material out of it, whether it's to go through inbreeding and extract good inbreds, or maybe they're not inbreds, but whatever the material is, you'll cross-pollinate plants from the two populations with each other and hopefully produce a superior hybrid variety by doing that. So again, the objective is hybrid variety production. Before I move off this slide, let me ask you then if there are any questions. Again, it looks complex, but it's a lot like what we saw before, and these two halves are mirror images of each other. Did you have a question there, Andres? Okay. Anything else? Anybody else before we move off? Okay. Advantages. People who have tried this in the past and reported it in the literature have found that not only, I'm starting with the second bullet in a sense, not only are you improving the cross between the two populations to identify material that will nick better to give you heterosis and a good hybrid variety, but they've also found that the two populations themselves have been improved. Okay, so if you kept them by themselves and looked at yield after 4 cycles of selection versus the base population, there's even been improvement in the population itself, let alone improvement in the cross between the two. So both of them have actually been accomplished in the past. Disadvantages by far compared to any other population improvement method that we've talked about. This one is by far the most work and the most complex to do it. You're essentially doubling the amount of work because you've got two populations you're working with at the same time. Again, it might be worth your effort to do it. People have done it in the past. You can do it in 1 year if you can get 2 seasons in a winter nursery. By the way, keep in mind, let me flip back just a couple of slides here. Let me ask you one more time by way of review. Which season do you actually do at your home base in contrast to the seasons that you can do in a winter nursery? Because it's very important to keep that in mind. Which one would you do at your home base where you're producing the variety for? Season 1, 2, or 3? 2, okay. Season 2, whichever season in any one of these methods where you're doing the evaluation for the trait, especially a trait like yield. Yield here in New York State is not going to be the same as yield down in Florida or Puerto Rico. You can do the seed production in Puerto Rico, but if you're going to evaluate for yield, then you need to do it wherever you're producing the variety for. And in this particular scheme, it's going to be season 2 for both of these populations where you're evaluating the half-sib progenies. Okay, so a very important concept to keep in mind on that. Okay, let me then do a little review and back up and look at the bird's eye view on this whole thing. Now we've covered all 10 of the population improvement methods, most of them being intra-population improvement and the last one being inter-population improvement. So let's use this time as review. First of all, to look at the biggest picture here in population improvement, we split it off into inter-population improvement, where we're working with two populations simultaneously, as we just have been talking about, for reciprocal recurrence selection. And it could be half-sib or full-sib, or there's all kinds of reciprocal recurrence selection. We just covered the one. But again, our objective was hybrid variety production. With the intra-population improvement, your goal is to improve one population at a time. Okay? Now you might still do some hybrid variety production, but you'll be working on different populations, keeping them separate, maybe trying to improve them for different traits, and maybe extracting material out of 2 or more populations to produce some kind of a hybrid. Or it could be any other kind of a variety, okay? It doesn't matter. But you're just improving a single population at a time. We split that down into our phenotypic methods, where you're evaluating individual plants. And then we talked about a whole bunch of genotypic methods that are the progeny tests. And we talked about 4 types of half-sib progeny tests, just modifications of one another. We talked about the full-sib method, the S1, and the S2 method. Okay, so there were quite a few of those genotypic methods that we discussed there. So that's the big picture of population improvement. Now let me take each one of these categories, especially the phenotypic and genotypic, and do a little review on that. Last week when we talked about the phenotypic methods, we started out by looking at recurrent mass selection. And recall that you're just growing out a bunch of plants, maybe a few thousand plants, identifying the individual plants or evaluating individual plants, and just harvesting the seed off of the superior plants to produce the cycle 1 there, cycle 1 seed. Now, that can be a really good method if you're evaluating the trait before pollination occurs so that you can control the pollination. You can only allow pollination among the selected plants so that you're selecting not only the female gamete but the male gamete. So it's effective for that. But for traits where your evaluation is after pollination occurs, for yield for instance, pollination already has to occur for yield to even happen, then it's not as effective. It can be effective, but theoretically half as effective in terms of progress from selection per cycle than you would if you could actually control pollination. Your seed that you're saving, it occurred because of pollen that came from the entire population, the non-selected as well as selected. So what did we do to modify that method so that we could be selecting the male as well as the female gamete? What did we do in a crop like corn to make that modification? Now this is, since you're not having an exam anymore in this class, this is our oral exam for the whole class to see if you can remember and understand the most important concepts that we've covered the last 2 weeks. Okay, how can we modify that in a crop like corn so that we can be selecting the male as well as the female gamete, even though we're evaluating the trait after pollination occurred? Andres? Okay, self-it. Okay, so you're not only producing open pollinated, one ear that's open pollinated so that you can evaluate yield, but you also have to self-it on another ear so that what you actually keep, what you save, is not the open pollinated seed. You're just using that as an evaluation for yield, but it's the S1 seed of those superior plants that you're going to keep. You grow those out the next generation and allow those to interpollinate so that you can get that genetic recombination. Hopefully get new recombinants so that the next cycle you can identify even better genotypes out of your plant population. Okay? Now how would we do that in a crop like alfalfa that's perennial? We don't necessarily have to produce S1 seed. What could we do with that, with a crop like that, to make a modification and accomplish the same thing? Anybody remember? Nick? Yep. You could dig up the plants. It's just like grass. Alfalfa will regrow again when you harvest it, when you chop off the top growth. So in the grasses, we would actually leave those in the field because it's a little bit harder to work with in the greenhouse because they're wind pollinated. It pollinate everything in the greenhouse and be hard to control the pollination when we've got several populations there. So we would keep them in the field, either in the same place, but then when the regrowth comes back, we would just harvest off the plants that we do not want to pollinate and only allow those that are superior to come up into the head stage and to pollinate with each other. Or we could dig them up and put them in an isolated field. Or with alfalfa, we do dig them up and bring them into the greenhouse. And then we put them on carts and roll them down to up there in Guterman, we roll them down to a room where we let the bees do the pollination for us. So in that way, with a perennial crop, we don't have to produce S1s. We can go back to the original parent plant and allow those to interpollinate with each other. So it's really a nice, slick way to work it. And that's called recurrent phenotypic selection or phenotypic recurrent selection. Doesn't matter too much the order of the adjectives there. By the way, are any of you in here, have any of you had any experience working with potato? Have you? Okay. With potato, what could you do with a crop like that? It's not perennial, it's annual, but what could you do as a modification and still not, I don't know if you can you even produce S1s on potato, or is it incompatible? Is it? Okay. So what could you do if you really wanted to do something like this? Okay. Vegetatively propagate it by the tuber. So even though the plant is annual and you can't do like alfalfa and dig it up and put it in a greenhouse, you can keep the tubers. And the tubers are going to be identical to the plant that they came from genetically, and so you can keep those. Can you keep those, if you keep them under dry and cool conditions, will they last more than a year?
Yeah. But what they do is
culture, and so they just grow them in little vials, and when they get to the colony, they cut up the berries and then put it back in the vial.
Oh, okay. So by doing that, you can keep them around for as long as you want, essentially. Okay. Okay, so, you know, that just illustrates the point that I brought out, I think probably the first day that I was with you 2 weeks ago, that although we're going through these methods and we're using corn as kind of the classical example, and I'm giving an alfalfa as an alternative for some of these, each crop is going to be a little bit different on how you handle it. So there's an example with potato. It's not an annual, but you can vegetatively propagate it. So there are different things that you can do with different crop species to modify these methods to work for your advantage. Okay? And those are the kinds of things that I really want you to know. You know, I don't expect you to be able to regurgitate what a particular breeding method is or anything like that, but if you know the principles that we're trying to cover, and I keep going over and over again with you, to be sure it's ingrained in your mind, then you'll be able to modify any one of these methods to your advantage in the future. Now, with any of these methods, we can use this stratification. Anybody remember what stratification is? Or another term for it is the grid system. Anybody describe briefly what that is? Yes, go ahead.
Control for variation within the field by where you plant the plants.
By what again?
By how you plant the plants.
Okay. Well, basically, it's the, I mean, not necessarily how you plant it, but how you do your selection. You know, if you have a lot of variability, soil variability in your field, which we do around here, you know, an extreme amount of variability compared to prairie soils out in the Midwest, that could really make it difficult for you to identify the superior genotypes. There might be one part of the field where the plants are growing a lot better than another part of the field. And if you just say, I'm going to select the top 10% of the plants out of the field in general, then you'll just end up selecting plants out of the best part of the field, and they're not necessarily genetically superior to plants that are in the worst part of the field. So you block off your field into different grids, you know, into several blocks, and instead of just saying I'm going to select the top 10% out of the whole field, now you say it's going to be the top 10% out of each individual block. And so the environmental variability becomes less in terms of the influence it has on your identifying the superior genotypes, because now it's just the environmental variability within a block, not over the whole field. And hopefully the variability within a block is a lot less. So you should be more successful at identifying superior genotypes within a block compared to over the entire field. You still consider it as one plant population. Once you have identified the superior plants, you still put them together and allow those to interpollinate as one plant population. Okay? But again, you're trying to improve the success of the whole breeding scheme. Now I'm going to write on the board here just a general, whoops, let me do it this way. A general equation for response from selection, like we started out with, is this I, the selection intensity, times A. Again, that little A factor has to do with whether or not you have pollination control and what kind of unit you're evaluating, whether it's individual plant or some kind of progeny, times the additive genetic variance, divided by the phenotypic standard deviation of whatever the unit is that you're basing your selection on. With the grid system, this is the part down here that you're affecting. You're trying to reduce this part to the extent that you're reducing the environmental variability, okay, on the influence it might have on your ability to select the superior genotypes. So this is the part that we're influencing. Anybody have any questions with that then? Okay, let me turn this off so we can go back to the slides then. Okay, so this is all phenotypic selection. We covered all of that last week. This week we were jumping into, in a real deep way, the genotypic selection methods. And again, there's a whole bunch of those. With the half-sib methods, we started out with the simplest one that was developed over a century ago, where you simply you're open pollinating the plants to produce half-sib progenies, evaluating the replicated half-sib progenies next season, taking remnant seed of those half-sibs and bulking that to be your cycle 1 seed. So relatively simple compared to some of the later methods. But with that, it had an A value of 1/4 because of the lack of pollination control and because you're working with a half-sib progeny that has less additive genetic variation among those units there. So it's 1/4. So then what becomes the advantage of using that method? What's the major advantage of the genotypic methods? Anybody remember? I mean, if you're reducing that A value down as much as 1/4, what advantage is it to use a genotypic method like a half-SIB progeny test in the first place? Why would you go through all that effort? Anybody? Yes? Okay. What you're trying to do is to evaluate plants that are at least related to each other. In fact, for the replicated clonal propagation, they're identical plants. But you're replicating them in micro and even macro environments so that you can have an evaluation in more than one microenvironment. It averages out the environmental effects so that more of the differences that you see when you're comparing the mean of one half-SIB progeny with another one and another one are more genetic than they are environmental, or at least less environmental. I mean, you never get rid of the total environmental effect with a method like this, but at least you reduce the effect that it has on your ability to select the superior genotypes. So again, like the grid system, it's this denominator that you're reducing. So the question comes, if you're reducing with these genotypic methods, you're reducing this A value compared to what you have with the phenotypic methods, then when does it become more of an advantage to use a genotypic method than a phenotypic method? And it has to do with the level of heritability. Which one would you use which? If you have traits of a fairly high heritability, medium to high heritability, which would you use, phenotypic or genotypic method? Okay, phenotypic method. If the phenotypic method will work, use it, okay? If you have medium to high heritability and phenotypic method works, use it because it's by far the simplest. You can evaluate a lot of plants, select a lot of plants, minimize inbreeding depression, conserve alleles for other traits because your population size is bigger. But if it doesn't work, if the heritability is too low for phenotypic selection to work, then you might have to resort to a genotypic selection method. And that's exactly what we have to do with a disease, as I mentioned last time, with spring black stem resistance on alfalfa. We've actually tested it, computed heritabilities for phenotypic and half-SIB selection, and found out the only way it's going to work is to use a genotypic method. So with the genotypic method, even though the A value is decreasing, if you can decrease this phenotypic standard deviation, and remember that this phenotypic standard deviation can be partitioned out into a genetic component and an environmental and also G by E. If you can take these two things and decrease the proportion of those that are part of this phenotypic standard deviation so that more of it becomes genetic compared to the environmental part of it. In other words, if you can decrease this part in the denominator by more than you've decreased the A value, then a genotypic method is going to be advantageous. Otherwise, the phenotypic method is going to be better because you've got a lot larger A value. The only time that this denominator is going to decrease more than you've decreased that A value is when you do have a low heritability, meaning that there's a lot of environmental variability for the expression of that trait in the population. And you're trying to minimize, as much as you can, the influence that environmental variability has on your ability to identify the superior genotypes. Let me pause and ask if you have any questions then, because that was a mouthful. Okay. Again, and for any of you who would be taking my quantitative genetics course next semester, we get into this equation in a lot more detail so that you can understand it a lot more thoroughly and really have a lot better concept as a plant breeder, what you as a plant breeder can do to be more successful at identifying the superior genotypes in a plant population. So we get into it in a lot more detail. Okay, let's go on then. So we had this original half-SIB method, but there was lack of pollination control. Then what kind of modification could we make with that with corn then so we can be selecting the male as well as the female gamete? Same answer as you gave for phenotypic selection. Okay, you could produce S1s on the parent plant. So that's what this modified half-SIB was. In corn, we combined the S1s. In alfalfa, go back to the original parent plants instead of using S1s. and potato, maybe go back to the original parent plants, but that's being stored in the form of a tuber or something that's in a culture, a test tube culture, okay? But it's essentially going to be some kind of a vegetative propagule. Okay, so again, there's different ways that you can modify this to be selecting the male as well as the female gamete. We talked about combining mass selection with half-sib selection with what corn breeders call ear-to-row-to-ear method, because you're producing an ear of seed that's essentially half-sib progeny, growing it out in a row to evaluate the half-sib progeny. Doing your mass selection, so you're coming right back to selecting an individual plant, which means that you're harvesting an ear of the grain to be your cycle 1 seed. So that was a combination of mass and half-sib. Anybody have questions on that? Yeah, Andres?
The question is about when you talked about comparing the A value between self-pollinated, when you were improving the method by knowing where the two gametes come from. Now, why is the A value the same for self-pollinated plant compared to the taking the same parent plant and use it as a pollinator?
Yeah.
Because the genetic combination within the genome of both is going to be different. There might be some similarities, but the combination that made this particular plant be successful, it's not completely going to be seen in the S1 progeny when compared to the parent plant.
That's an excellent question. Part of the answer, I mean, I'm not sure I can actually repeat the whole question that you asked, but it essentially had to do with why is the A value 1 for the S1 progeny method, and it's essentially the same as some of the other methods where you might be recombining S1s, or maybe even compared to a phenotypic method that was 1. Something like that? Okay. Is that close enough? Okay. I mean, if I'm not answering correctly, then ask it again if you could. But essentially what's happening with the S1 method is that there's two things there. Number one is when you go through the S1 method of selection, you are selecting the male as well as the female gamete because there's no open pollination at all at that point. You know, you're taking the remnant S1 seed of the superior S1 progenies and recombining those. So you're selecting the female and male gamete there. So that's one reason why the A value is 1. The other reason has to do with the type of progeny that you're evaluating, in the sense that if you have like 3 parent plants and you're crossing those in all combinations and you're producing half-sib progenies with them, then the distinctiveness among the half-sib progenies, let me see if I can illustrate this on the blackboard here. If you were to look at a curve in a sense, and you had a performance, let's say the farther to the right that you get, the better the performance it is for a trait like yield. And so I have individual plants and you know, you've got a plant here and a plant there and so forth. But anyway, they're distinct plants in performance for that particular trait. Once you take that plant and you just allow open pollination and you produce half-sib progenies, what's going to happen is that theoretically, probably not quite true in practice, but theoretically, you've got a mixture of pollen from all over the population, and that's the same pollen that's pollinating each individual plant, so that the progeny are going to be distinct only by the genetics, the distinctiveness of the female parent, but the pollen that produced that seed for the next generation is the same. Okay, so every half-sib progeny has about the same pollen that pollinated it, theoretically. Again, in practice, it's probably not quite that true, but I think you probably can get the gist. So the distinctiveness is only among the female gametes there. So what's going to happen is that you've got a lot of genetic variability within a half-sib progeny, as we talked about a few times ago. And so now instead of distinct plants genetically, now the progenies, a half-sib progeny looks like this, and they overlap with each other. Okay, like that. So there's less distinctiveness. There's less genetic variability among half-sib progenies as there was among the parent plants. Does everybody follow that? Okay, so that's one of the disadvantages of a half-sib progeny test. With a full sib, there's less genetic variability within a full sib because now it's only cross-pollination of 2 of them at a time instead of pollen from all over the population. So this full sib progeny right here compared to that full sib progeny might not even be related to each other because there are 2 different plants that were used for crosses of those full sibs. So there's going to be more genetic variability among the full sib progenies. Even more so among the S1s, because now the S1s, it's going to be harder for me to draw it, but there's going to be some genetic variability within an S1 progeny, especially if the parent plant is heterozygous, and then it's going to, by self-pollinating, it's going to segregate out. So there's going to be some there, but it's not going to be as much as for a half sib, and there's still going to be more distinctiveness there among the S1 progenies because there's less genetic variability among plants within an S1 progeny when you self-pollinate it compared to cross-pollinating it with a whole bunch of plants over the whole field there. Is that starting to gel with you? Is sort of?
My question is more of the genetic combination. By doing a self and using those seeds to open pollinate with the population, those seeds of the self plant were segregated. The genes were
Right.
segregated. So the possible combination that made it successful in the first plant, in the parent plant, is not going to be seen as much in the part.
Okay. So you're saying if you have a really good combination of genes in a parent plant, you might lose it when you go down and take S1s and cross it. That's possible. It could happen, which would be very important if you were right at the very final stage of producing a variety, especially a hybrid variety, where you want to make sure you get a really good combination of genes right at the end. So it is possible that you could lose it, but through population improvement, when you're going through multiple cycles of selection, in other words, recurrent selection, your objective through that is to mainly just to increase the gene frequency of the favorable genes, favorable alleles in the populations, and then try to get good combinations right there near the end or at the end to produce your variety of whatever sort it might be. So yeah, you could lose a good combination, and that is one of the hazards of it. If you feel you have such a good combination of genes to start with, then that might be a set of plants that you might want to use as parents for a variety right there at that time. But if you think that you can get better recombinants, which is what your objective is through population improvement, increase the frequency of favorable alleles and get better recombinants. What you hope to do is to cross-pollinate populations or not populations, but plants within a population, because this plant over here might have a good combination of genes, and this one over here might have a good combination too, but it might be for different reasons. It might be a different set of genes that make them good. If you can cross-pollinate those together and hopefully in the progenies get a better combination of all of those genes into the same plants, that's kind of what you're trying to do with population improvement. To build up the frequency of favorable alleles, give you a better chance of getting better recombinants by cross-pollination as you have a higher frequency of favorable alleles, and in the end try to get even better combinations of genes than you had started with. Does that help better? Is that kind of, I'm not sure I'm still answering your question, but any other questions on this? Okay, let me go on and then we'll just finish up here in just a moment or two. Okay, so we had this mass selection combined with half-sib selection. We also use this half-sib progeny test using a tester, with our example being a poor-performing tester where we're trying to distinguish between homozygous dominant and heterozygote. This is not a method you probably would use a lot. You could probably go through other selection methods and get a long ways with that, even if you have complete dominance for a trait. But if you hit a plateau, which would happen if you can't distinguish between homozygous dominant and heterozygote, if you felt that you needed to improve the population even more, then you could resort to this method or molecular techniques to identify superior genotypes there. The full SIB method, what was the advantage of that then compared to the half SIB methods? Remember, the full SIB method has even more genetic variability among those progenies than the half SIB, so your A value doubles to 1/2. The S1 even more, as I was illustrating kind of in a sloppy way on the board here. So your A value for the S1 method is going to be 1. The S2 method, you actually end up with 1.5 for an A value. Because you're self-pollinating them, you're getting more homozygosity with the plants within a progeny row, more distinctiveness among the progeny rows there. So even more and more of an A value, a higher A value as you go along with these methods. But again, it gets more and more complex, and the S2 method actually takes longer to accomplish. Okay, let me end with this next series of slides here. Now that we've talked about all of these methods, how do you decide which one or ones to use? And you might decide to use different ones for different plant populations or different species. But I use the acronym THEO, which is the Greek word for God, or Theodore, or theology, or however you want to remember it. If you were taking an exam, it would be a nice acrostic to try to remember if somebody were to ask you the question. But since you don't have an exam, you don't have to worry about it. But basically, theoretical gain from selection, the response equation that we looked at the A value a lot, but even talked about the phenotypic standard deviation in the denominator for the genotypic methods and clonal evaluation and the grid system. Okay? But comparing the theoretical gain from selection can help you with that as well. When you do that, you need to be looking at it per year's time. We just looked at it per cycle, but if it takes 2 years to accomplish something versus another method with 1 year, that weighs pretty heavily. Okay? Heritability, we already said by way of review today that if you have a trait with medium to high heritability, that the phenotypic methods work well. If the trait has a really low heritability, like less than 20%, then you might have to resort to using a genotypic method, a progeny test. The amount of work that it takes, ease of selection. Phenotypic methods in general are a lot easier to accomplish than the genotypic methods, but even with the phenotypic and within the genotypic, there's variability on the amount of work that it takes. So you have to consider the resources that you have, what you can accomplish. And the objective, primarily I'm thinking of if you are planning to produce a hybrid variety for that crop species, then you could use reciprocal recurrence selection. You wouldn't have to, but you could use it. If you are not producing a hybrid variety, then it's kind of a waste of time to use reciprocal recurrence selection because it's a lot easier and just as effective, if not more effective, to use one of the intra-population improvement methods. Anybody have any one last question or questions?